In this video, we will discuss about how this TGF-beta triggered apoptosis occurs, whether it depends on how the cells spread or it depends on what kind of ECM is used. So the initial hypothesis that they have is that whether this TGF-beta triggered apoptosis is because of the specific kind of ECM that is used on the compliant hydrogel. So what it did is they first checked different kind of ECMs, and they have tested fibronectin, recombinant basement membrane, or commercially it is known as Matrigel, and this collagen 1. So in this case, what they are able to see that in the case of compliant, and these results for the panel A is after this TGF-beta treatment. For the compliant, for the fibronectin and recombinant base membrane, they do not see cell spreadness, but for the collagen 1, they are able to see cells are able to spread to some extent. And thereafter, and similar results they have seen for the rigid also. Then they checked whether this ECM, different kind of ECM affects the EMT changes, that is epithelial-to-mesenchymal transition or not. So for that they checked the snail-1 mRNA expressions, and for the case of this fibronectin and recombinant basement membrane, they see the expected results, that is for the rigid case the ECM for rigid case, and in the TGF-beta conditions ECM is significantly higher compared to the control. But for the compliant case, it looks like it increased, but this increase is not statistically significant. And similar results they have seen for the collagen ones in the rigid case and for the compliant case also. That is, ECM does not get affected because of the change in the EMT does not get affected because of the change in the ECM. Now there comes the main question of their hypothesis, that is whether this different kind of ECM affects the apoptosis or not. So in the case of fibronectin, they are able to see increased apoptosis in the compliant case, and similarly for the case of Matrigel, they are able to see increased apoptosis in the compliant case, and that too in the TGF-beta treated conditions. However, for the collagen 1, they see slightly increase, but an increase is not statistically significant, and for the rigid there is increase is even lower. So from that, the one thing that comes to their mind is that it could be the cell spreadness, that is that might plays a role in inhibiting apoptosis. So in order to check that, what they did is they did the microcontact printing of all the 3 ECM to prevent the cell spreading, and then check the caspase-3 expressions on the compliant cells. So this is the representation how it looks like. When there is a microcontact printed, the cell spread is not there, whereas when there is a complete coating of this hydrogel is there, they are able to see cell spread clearly. So now they check the this apoptosis markers, that is caspase-3 positive cells, and they are able to see in the unspread conditions the caspase-3 expression levels are significantly higher compared to the case of the spread conditions. And however, one more thing that they have observed, that apart from the spreadness that helps in inhibiting TGF-beta triggered apoptosis, they see that the collagen 1 has significantly lower apoptosis even in the unspread conditions compared to the fibronectin and recombinant basement membrane or the Matrigel. So they also made one more point, that collagen, apart from the cell spreadness, this collagen 1 also plays a role towards inhibiting this TGF-beta triggered apoptosis. As we can see that the signals that are seen for the percentage caspase-3 positive cells is significantly lower compared to the fibronectin and recombinant basement membrane TGF-beta treated compliant unspread cells. So from that they made a point that it's not just the cell spreadness. Cell spreadness plays a role towards inhibiting apoptosis, but the ECM2, this collagen 1, so there is a error, it's collagen 1. The collagen 1 also plays a role towards inhibiting TGF-beta triggered apoptosis. This is what they finally conclude, or finally come to the point from the results that are presented in this figure. If you need to understand more about these experiments in detail and wants to refer to the supplemental figures, You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.